CTL, cytotoxic T lymphocytes, are natural effectors of our immunity. They, in this case, are capable of specifically killing cancer cells. These cancer cells are shown in these yellow boxes. Further on, the CTLs will appear in red boxes, and as you can see on this picture, the cytotoxic T lymphocytes has been able to recognize specifically a tumor of the mouse DBA2. This tumor is a Mastocytoma P815, and this is why we called this clone of CTL T lymphocytes P8915. As you can see, amazingly enough, these cells move about by reptation very closely that movement which amoebas use. Here you see these cells, after killing cancer cells, very near the threshold of the medium and the dry. In this case, we see on the bottom part a cancer cells, which is bigger, surrounded by two killer cells, which have struck her after recognizing the receptors. The killing is taking part now, and the cancer cell is fighting back by pseudopodic activity, trying to get loose of their well-tight killers. You can see on the top part where two other cells lie that a cancer cell already is barely only a ghost. As you can see, the lymphocytes on the right part of the bottom image has probably been the effector killer. After recognizing the receptor, the killer cell strikes it again with another part of its membrane and injects doses of a protein called perforin, which makes a hole in the cell, allowing not only toxins to flow through, but also an osmotic destruction of the cell, which you can see here, growing thicker and ghosts. Now here we see only one killer cell left, and one of them is leaving the target, having done its job and ready to strike again, while the other cytotoxic T lymphocyte has a very difficult time in getting away from its target after the kill. This is a very unique phenomena that has never been recorded before. You see on the right a randomly going CTL. Indeed, CTLs do not have any apparent chemotactic gradients recognition, and they strike their target, their very specific target, which is the P815 mastocytoma, at a random. As you can see on this image, after the kill, which took about 20 to 25 minutes, it has been now up to 4 or 5 hours in which the effector cells has not been able to get rid of the cadaver of the target cell. This, of course, is an impairment of productivity and performance, which we can see even in the most perfectible biological system can happen. Here we see that at last the effector cells, the CTL, has been able to get the target cell away from the surface and is slowly able to take away the tail end of its body from the hole that it had made in the target cell membrane. Yet it apparently cannot get completely free of it, even though it is now totally disconnected from it. Here we see one of its compatriots coming about the scene of the action, and apparently through the movements and gradient of turbulence, the cadaver, the ghost, will leave the scene, leaving thus the effector cell. You see the cancer cell moving, the dead body moving away and out of the focus of the picture, and the other effectors capable of another action. On the top part you will see a very interesting phenomena, which is the mitosis of an effector cells. Indeed, CTL, through the monoclonal proliferation, are proliferative cells which enables us to keep these clones and to produce vast amounts of them. But in this particular case, we almost lose these dividing cells. A very striking and apparently marginal, never described before phenomenon appears. As you can see, the cells are well polarized. We see definitely two nuclei on both ends of the cells, which are linked by a very long septum, which does not break, however. So we are there in presence of definitely two entities linked again, and who are trying their best to, as nature calls for, pull apart, while on the left part of the picture you see a mitosis that has been accomplished, and in which the cells will migrate from each other and will become two different effector cells, CTLs. Now back in the middle of the picture, however, we see that the once polarized dividing cells has apparently fusioned again, completely reuniting their two nuclei, destroying the microtubular system, and we shall never know if it will divide again, because it is leaving us. Indeed, these cells are seated on cover slips, and we can never know exactly before we start an experiment what will happen in the time lapse. As you see here, this is to show you what time lapse can give. This is a static image which turns into an active image of video time lapse cinematography, which has enabled us to vision directly many strange and unaccounted for phenomenon of the biological interaction and of cellular events, which we hope will enable us to explain more of the mysteries of the cellular biology.